Urine formation. There are three processes involved in urine formation. First is glomerular filtration. As the blood flows through the glomerulus, it contains both useful chemicals and dissolved waste products, which are soaked from the blood through membranes by osmosis and diffusion, where it is filtered and then flows into the Bowman's capsule. This process is called glomerular filtration. It consists of water, salt, glucose, and other chemicals. The normal glomerular filtration rate, GFR, in an average-sized man is about 125 milliliter per minute. Second is tubular reabsorption. It is the movement of solutes and water back into the bloodstream. It changes the composition of glomerular filtrate and reduces its volume markedly. And the third is tubular secretion. In secretion, substances move out of the blood into the tubules, where it mixes with water and other wastes, and then they are converted into urine. Function of the tubules. Proximal convoluted tubule PCT. The proximal tubular cells are characterized by the presence of brush border on their luminal surfaces. The brush is composed of thousands of villi, which increases the absorptive surface area of each cell by 20 folds. The proximal tubules reabsorbs approximately 67% of the filtered water, Na+, Cl-, K+, and other solutes. Almost all the glucose and amino acids filtered by the glomerulus. The proximal tubule secretes H+, urate, penicillin, sulfonamides, and creatinine. Function of the tubules. Henle's loop. About 20% of the filtered Na+ and Cl-, 15% of filtered water and cations such as K+, Ca2+, and Mg2+ are reabsorbed in the loop of Henle. In thin descending limb of loop of Henle, water is absorbed passively and it is accompanied by diffusion of sodium ions from interstitial fluid into tubular lumen. In thin ascending limb of loop of Henle, limited passive reabsorption of Na+ and Cl- occurs in this water impermeable limb. Because of impermeability to water, the fluid leaving this limb is hypotonic relative to plasma. Distal convoluted tubule DCT. In the initial segment of distal tubule, it reabsorbs Na+, Cl- and HCO3- and secretes hydrogen and potassium ions. Collecting duct. It reabsorbs water to produce concentrated urine. Mechanism of concentration of the filtrate. A counter current system refers to a system in which the inflow runs parallel to, counter to, and in close proximity to the outflow of some distance. The counter current flow system is formed by U-shaped tubules. In kidney, loop of Henle and vasa recta form the counter current system. The close proximity of loop of Henle and vasa recta is responsible for production of hyperosmolarity and a gradient in the renal medulla. This gradient is caused by NaCl and urea. The ascending limb of the loop of Henle transports NaCl and then exchanges it with the descending limb of the vasa recta. This is called the single effect. Due to multiplication of single effect, the osmolarity of interstitium rises progressively from cortex to medulla and papilla. Urea plays an important role in the development of medullary osmotic gradient. Countercurrent exchange of solutes between ascending and descending vasa recta minimizes solute washout from the medullary interstitium. This transport of substances is called counter current mechanism.